So yeah, once again, so I'm from the computer vision and the deep learning field, but in this talk, I will relate medical imaging to biology. There might be some takeaways at the end that can help biological studies by using the similar method. So to start with, I want to briefly talk about the relationship between the medical imaging and the biological process. So in this example, the goal here is to detect a lung tumor from CT scan. As the figure shows, this patient CT scan goes through a 4-stage process to determine if there is a tumor. So this process includes identifying ROI and segmentation, and render this into 3D, followed by the feature extraction, usually radiomic features. And finally, we apply some statistical analysis to determine the malignancy. So what is the radiomics and how robust are they? So radiomics is a category of handcrafted imaging features that are designed to make a distinct characteristic between the disease forms. So they have the potential to uncover disease characteristics that are failed to be appreciated by the naked eye. So there are like first order shape, like for first order feature, like the shape feature, for example, the volume and texture features. So the recent availability of a large CT dataset with advanced in medical imaging analysis enables a growing number of machine learning methods that utilize radiomic features for prediction and classification. So however, as you can imagine, a few are used in this clinical practice. One significant barrier is the poor reproducibility for radiomics features. So this is largely due to the variation in how CT scans are acquired and reconstructed has an impact on the outcome. So this figure shows on the right, the ICC intra-class correlation coefficient. So although the shape feature here, let me show you. Okay, although the shape feature highly reproducible, but the texture-based features such as GLCM are not so much reproducible and have very high variabilities. So here, another example is that we applied a similar analysis to our in-house data set and showing that radiomics are not very reproducible. So in this figure, we look at the heat map of concordance correlation coefficient for various conditions on some representative radiomic features that are proven to associate with nodule malignancy. And we can see many of these entries have poor reproducibility as indicated by red color, and especially on the GLCM texture features. So we need to be cautious when we're working on these radiomic features. And so what are those parameters that can have an impact on radiomic features? For example, the first one is the dose. The doctor may adjust the dose level based on patient's medical information. For example, the figure on the left is low dose CT screening, and on the right is follow-up diagnostic CT on the same patient. You can see the noise level are very different. And the second thing is, during the acquisition, CT images are reconstructed from raw data or the spatial domain to image domain. This due to different tissue attenuation and contrast, we need to apply different kernels and filters to reconstruct the image to look at different organs. For example, here this is a sharp filter, a sharp kernel which resulted in image in this appearance, and here I'm showing a smooth kernel, and the image is kind of blurry. So they have very different appearances. And the last thing is the technician may choose speed over resolution, so resulting poor Z resolution, which eventually that can miss small lesions. So in order to study the effect of various conditions, we need a massive amount of CT data that have a different acquisition and reconstruction parameters. This is not easy to obtain such a data set. So instead, we have an accurate way to generate various of reconstructed CT from raw projection data and obtain under different acquisition settings. So from the acquisition of 186 UCLA low-dose CT screening chest studies, a physics-based noise model was used to simulate the reduced dose sonograms. And those sonograms were later reconstructed into CT images using weighted filter back projection with a choice of kernels and the slice thicknesses. So together we have a variety of acquisition and reconstruction conditions. In total, there are 36. So those example reconstructed images are shown in this figure here. Okay. So next, our goal is to normalize those various imaging conditions to a reference. For example, as shown here. So the same image, so the same CT image reconstructed under 36 conditions is shown in this table. And all have very different appearances, as you can see. The underlying assumption or the hypothesis is that if we can make the appearance, the image quality, alike to each other, then the radiomic feature can be improved or more reproducible. So this is well known as the image translation in computer vision society. There have been plenty of applications in the medical imaging domain. For example, converting a low-dose CT image to a denoised version, or do some super resolution, and synthesize some medical images, like from the vessel to eye fundus, or even the cross-modality transfer from CT to MRI. So one baseline method that I'm showing here is using the CNN, which stands for the convolution neural network in deep learning. And here we train a 3D CNN model to convert a noisy 10% dose image to a 100% dose image using multi-layer of residue connection network. So after the network learn the mapping, we take the image from the test set, and here's the result. If you zoom in, you can see it does a decent job in terms of denoising, but however, if you look closely, it's kind of blurry and some important detail like textures are missing. So here is another animation in the along the longitudinal direction. So you can see that does a decent job, but it's not very realistic due to blurred textures. Those textures are actually very important to determine the lesion from those morphological details. So how can we generate more realistic images? So one powerful method is called generative adversarial network, or GAN. So this method has been drastically improving over the years. For example, the quality of synthesized faces becoming more and more realistic. So GAN consists of two networks, a generator and a discriminator. So take the handwritten digit generation for an example. So during the training process, the generator tried best to generate a realistic digit, where the discriminator job is to distinguish if the generated numbers looks real or not. So generator tried best to fool the discriminator, and this adversarial training keeps going to learn the true data distribution. So during the inference, we ditch the discriminator and only keep the generator for the image generation. And there are very interesting applications come out of this. For example, in the domain transfer, in the natural image domain, such as turning a horse to a zebra. So here we use a generator that has the same network structure to the previous CNN-based network. And the discriminator, which acts like a binary classifier, will tell you how realistic the normalized the output compared to the ground truth. So if the discriminator found out that the output of the generator is not very real, this piece of information will backpropagate to the generator to update the weight, its network weight accordingly. So for the next iteration, you will generate something more realistic. So however, as you can imagine that GAN is very difficult to train due to the adversarial nature. There are two networks fighting against each other constantly. And so here we also use a unique spectral norm layer in the discriminator network to stabilize the training. And here, next slide, I'm showing some results. So on the left is the input scan, and on the right is the GAN output. As you can see, the image is much clear and has good texture information. And comparing the CNN result with the GAN output, you can see CNN is quite blurry, but GAN has keep the good textures. And we can also compare, look at the coronal plane. The figure on the left is the input, and the middle one is GAN, and the one on the right is the ground truth image. Like, as you can see, the GAN and the ground truth, they look very similar. Sometimes it's very hard to tell, like even for myself, like which one is from the GAN, which one is from the ground truth. So we have seen the mapping by GAN from one pool image condition to the reference. But it turns out that we can also apply the same technique to many mapping scenarios. So here, 3 independent models are trained on like 10%, 25%, and 50% dose, 2 millimeter slice thickness to the reference condition at 1 millimeter. And they all look good and have very good appearances. So this method is very powerful. So what do we mean by like the result like looking good? Is it helping the image features or becoming more reproducible? So this is a question has not been answered. So we went ahead and conduct 3 evaluations on the image quality, radiomic features, and nodule detection task. So first of all, in terms of the image quality metrics, we use a learned perceptual image patch similarity to measure the image quality. As you can tell from the name, it measures perceptual quality, and it correlates well with our human visual system. And we also include the traditional PSNR, which stands for peak signal to noise ratio, and SSIM, which stands for the structure similarity. So those 2 metrics are the traditional imaging metrics. So a lower LPIPS is better, meaning it's good perceptual quality. So in this table indicates that the image generated by GAN has the closest perceptual distance to the reference condition. So which once again confirm that our observation that visually they look similar. So on average, comparing to CNN, the GAN method improved the perceptual similarity by like 35, 29, and 25% for each of 3 normalization scenarios, as you can see highlighted by this green box here. So next, we look at the radiomic feature and improvement. So in this case, we extract a nodule in the volume of interest, which is a 32 by 64 by 64 pixel region, and compute the radiomics in this region. Then we compare the error to the reference condition. We use the Wilcoxon rank test to determine if the error is significantly smaller to the unnormalized original input. And for this particular nodule, in this example, the error from the GAN, which highlighted, which is in this green box plot, becomes significantly lower after the normalization. And interesting finding is that CNN, which is in this white color, CNN even makes the error larger for some, like for the GLCM cases. But our GAN achieves the lowest error. So we just see a few number of radiomic features, but how about like look at many radiomic features? So we selected in total 1,008 radiomic features and combinations to study further. But as you can imagine, it's difficult to visualize 1,008 radiomic features. So instead, we use a t-SNE method to reduce the dimensionality into 2. And each plot here represents 1 mapping scenario. If I zoom in here, if I zoom in here, as you can see, GAN is very powerful, bringing the distribution back to the reference. Where CNN like actually does no good in the radiomic feature for most of the cases. As you can see, the reference condition is in the reference condition distribution is in red, and the GAN is in green. And as you can see, the CNN is in like orange, but for most of these cases, scenarios here, CNN like even moves the distribution further away from the reference. But however, GAN can always align pretty good to the original like distribution here, the green and the blue one. Okay. So at last, in terms of the clinical task in automated nodule detection, we are facing challenges to distinguish the benign nodules from the malignant ones. It remains challenging to determine the individual probabilities of disease given the history findings on CT. For automated nodule detection, evaluating the risk of a patient with the lung cancer, low-dose CT screening, and identify those individuals who and when needed intervention is important. So this could reduce the morbidity associated with unnecessary interventions, or improve the survival rate with necessary biopsy. So all those challenges boils down to the early and accurate detection and prediction. So the next we evaluate on the nodule detection task on the F1 score. So we use a CAD system to perform those evaluations. So the CAD system, or the computer-aided detections, is one of the several approaches to propose to detect the nodule, lung nodules. So after the CAD detection, we compute the F1 score at subject level. And the goal here is that we want the detection performance of the lower resolution scans to be like the reference one. And the metrics we used is a concordance correlation coefficient, which just measures the agreement of the measurements comparing to the golden standard. And it turns out that our GAN-based method indeed makes the detection task more reproducible to the reference. So we can see that CCC improved comparing to the unnormalized scans, either in 2 millimeter, and also it's higher than the CNN results highlighted in green here, and also in the average. So in summary, in this work, we propose the 3D GAN approach to normalize heterogeneous images. It retains the perceptual characteristic of the target image or the reference. And we also optimize the model training, which making more stable and easier to train. And this facilitates more consistent performance on ML models by mitigating the heterogeneity in the data, in the input data. And so as an extension to our method, we can easily see that this GAN approach can be adapted into many other imaging types, such as optical image, MRI, or PET, and ultrasound. So it's not only limited to the CT modality here. And so one example of this is also the work done by UCLA. So they apply the super resolution approach, based on deep learning method, to fluorescence microscopy, and achieved pretty good results. So this is just one example in the optical image domain. And another extension that it can be useful to improve the robustness, where the variability in the imaging data has a strong impact on the biological correlates. Like, for example, the radiogenomics. So next, some limitation and the future directions. So GAN, there's one limitation of GAN is that it's possible that GAN can introduce some artifact in the generated or normalized image. So those artifacts might not be real in the original image domain or the ground truth. So we need to be careful with that. And another problem is that the super resolution, this type of problem is inherently a near post problem, which means many solutions are possible. And the next one is we need to train individual models for each of the new mapping, each imaging conditions to the reference. So this is not very efficient. So some future directions we are currently thinking about to explore is that we can so to basically to have a like stochastic alternative to the GAN to address the limitation, to address the second limitation, and also we will also explore a conditional GAN to incorporate the textual information during the training to create a unified model for all mapping scenarios to make it more efficient. So it's not necessary to train all the individual models for each mapping. So this, I think this is my last slide. And thank you. Thank you. And is there any questions?
Any question, anyone? All right, then. Thank you very much. And have a great weekend, everyone. Bye.
Bye.